Without further ado, I'd like to welcome my friend and colleague, Dr. Detlev Boison. He's currently at the Brain Health Institute at Rutgers University. And we've been collaborating on adenosine-based projects for a number of years. And right now we have a project going on in collaboration with Professor Harry Blaze and Professor Dave Ruskin here. And Detlev's been doing a lot of exciting and really transformative work on adenosine for a number of years, and I'm delighted that he moved to the East Coast a couple of years ago. And without taking up any more of his time, I'd like to really, let's give him a real round of applause and welcome Detlev.
Yeah, thank you very much, Susan, for inviting me to present today. And it's such a pity that I can't come in person. I've been looking so much forward to paying you a visit since I moved to New Brunswick last year in April. But then the pandemic struck, and I've known Susan and admired her work for basically a quarter of my own life. And we've been collaborating for a long time. And what I love about Susan, she is always so positive and inspiring in ideas and moving the field forward. And today I will talk about our favorite molecule, which of course is adenosine. So the title of my talk is called Translational Adenosine Research, and I will try to cover a broad range of activities ranging from epilepsy to cancer. So over the years, I've built a translational research program, all centered on the molecule adenosine. So we have a basic research arm where we work with transgenic models, animal models of disease to study epilepsy, traumatic brain injuries, the comorbidities of epilepsy, including SUDEP. Then we have a second arm of research directly related to therapy development. We work on gene therapies, stem cell therapies, on diet and metabolic therapies, and have launched a new drug discovery program. And then we combine everything into truly translational research in the areas of epilepsy, stroke, TBI, Parkinson's, Alzheimer's, brain repair, neurogenesis, schizophrenia, autism, and cancer. So that's quite a lot. So why can you build an entire research program around just one molecule? So the answer lies hidden in early evolution on our planet. So in the 1960s, smart people like Urey and Miller tried to figure out how life could form in the primordial atmosphere and the primordial soup of our early planet. And one of the key findings was that the toxic gas hydrogen cyanide can spontaneously form adenine. So adenine is nothing else but H5C5N5. So therefore, it's fair to assume that adenosine has been there right at the beginning of life. And therefore, it's not a surprise that you find adenosine everywhere. So adenosine is well known as a ligand to the G protein coupled adenosine receptors, A1, A2A, A2B, and A3. But it's also a core compound of a variety of metabolites, including ATP, ADP, AMP, SAM, SAH, NAD, FAD, coenzyme A, and of course, RNA. Adenosine is part of RNA, not DNA, and life started with RNA, and messenger RNAs have poly-A tails. So adenosine is kind of everywhere, and this makes sense based on its early invention in chemical evolution. So what is the principle of adenosine? So if you want to construct a living system out of scratch that is self-regulatory, you probably don't work with sophisticated protein kinase pathways and transcription factors and So you need very basic adaptive mechanisms. And the simplest system you can think of is if you have the energy molecule ATP and you get a depletion in ATP, you get the degradation product adenosine, and then select adenosine just globally to suppress everything that consumes energy. So this situation happens as a consequence of lack of food, lack of oxygen, or excessive energy consumption. And once you have too much adenosine in your brain, you end up like this guy. And what you do then is to think about caffeine, because caffeine is an adenosine receptor antagonist and helps you to get more alert. So adenosine in the brain has a lot of different actions. So the main actions are mediated by adenosine A1 and A2A receptors. In general, the A1 receptors are coupled to inhibitory G proteins and block neuronal transmission through blocking intake of calcium into the presynaptic neuron, which leads to presynaptic inhibition, and by promoting potassium fluxes on the postsynaptic side, which leads to a stabilization of the postsynaptic membrane potential and make postsynaptic neurons less excited. So this is a very powerful excitation dampening effect of adenosine. On the other hand, the A2A receptors basically do the opposites. They have excitatory functions in the brain and act more on a local level. In the brain, adenosine levels are largely under the control of adenosine kinase, which is primarily expressed in astrocytes. So astrocytes form a metabolic sink for the removal of adenosine. So adenosine is implicated in a wide range of pathological conditions, which are all listed here. And today I will focus on epilepsy and cancer. Adenosine is also an FDA-approved drug, which is given clinically for the treatment of supraventricular tachycardia, which I always consider to be a seizure of the heart. And our neurosurgeons use adenosine in the clinic. So when they do brain surgery and they encounter excessive bleeding, they don't see anything. They can't continue with the surgery because everything is just flooded with blood, which they can't stop. And they don't know which artery to clip in order to stop the bleeding. So what our neurosurgeons then do is to give an intravenous injection of adenosine, which immediately stops the heart. As soon as the heart stops, the bleeding stops, and then they know which artery to clip, and then they can resume the surgeries. So luckily, the heart normally starts beating again after a delay of a couple of seconds. So this is because the metabolic half-life of adenosine in the circulation is extremely short. So it's a very fast-acting agent to suppress cardiac function with a very short half-life, so that cardiovascular functions are restored almost immediately afterwards. So if we look into epilepsy, we are faced with a condition that affects about 1% of the population. Epilepsy is clinically characterized by seizures, which can manifest as altered perceptions of alertness, or so-called absence seizures, but they can also manifest in convulsive seizures, which can have clonic and tonic components. Clinically, seizures are diagnosed by electroencephalograms or EEGs, and brain areas that can be the origin of epileptic seizures can sometimes be visualized by MRI imaging. So the major problem of epilepsy is that about 35% of all cases are drug resistant. So that means that none of the existing anti-epileptic drugs works to treat those seizures. In addition, patients suffering from epilepsy are faced with comorbidities, which can be depression, cognitive impairment, or psychiatric disturbances. And persons with epilepsy are at an increased likelihood of sudden death, a phenomenon called as sudden unexpected death in epilepsy. So the problem is that none of the anti-epileptic drugs currently used in the clinic have been designed to prevent or to modify the disease. They are strictly symptomatic drugs aimed at suppressing neuronal excitability. So if you look at the mechanisms of actions of conventional anti-seizure drugs, which target epileptic hyperexcitability, you can see that although they have different targets, such as calcium channels, potassium channels, sodium channels, chloride channels, they basically all do the same thing. They just block neuronal transmission. Which is a relatively unspecific effect, which at the same time can explain profound sedative side effects triggered by those anti-seizure drugs. But the thing is that all those strategies, there's so many different targets, just don't work in 35% of all patients. So that means we need alternatives for treatment. And this is a very nice figure from one of Susan Sino's papers, which summarizes the effects of a ketogenic diet. A ketogenic diet is a high-fat, low-carbohydrate diet, which has multiple mechanisms of action. And that's a unique difference from conventional anti-epileptic drugs, which have usually just one or a few very well-defined therapeutic targets. So a ketogenic diet acts through multiple different mechanisms, which all combine to provide a potent anticonvulsant action, and also to have anti-epileptogenic effect, which means a ketogenic diet is able to modify the progression of the disease. And in one of our earlier collaborative studies with Susan, we discovered that one of the mechanisms of action of a ketogenic diet is the increase in adenosine, increased activation of adenosine A1 receptors, which is a study we published in the Journal of Clinical Investigation. So this basically means that a metabolic therapy is able to directly affect adenosine metabolism in the brain. So let's look at the biochemistry of temporal lobe epilepsy, which is one of the most frequent forms of adulthood epilepsy and which tends to be pharmacoresistant. And when seizures cannot be controlled with anti-seizure drugs, there is a surgical option for some of those patients where the epileptogenic brain area in the hippocampal formation is surgically resected. So if we look at the metabolism of adenosine, first of all, it's important to note that extracellular and intracellular adenosine levels are rapidly equilibrated through nucleoside transporters. So that means the intracellular metabolism of adenosine can affect extracellular adenosine and adenosine receptor activation. On the intracellular side, adenosine is linked to the phosphorylated compounds, the energy pool of the cell, to ATP. But it's also linked to biochemical transmethylation reactions, which include DNA methylation. So methyl groups are donated by S-adenosyl methionine, which transfers a methyl group, for example, to DNA, resulting in the formation of S-adenosyl homocysteine, which is then cleaved into adenosine and homocysteine. So what happens in the epileptic brain is that we find a pathological overexpression of the adenosine-removing enzyme adenosine kinase. So if adenosine kinase levels increase, we get a metabolic sink of adenosine, which drives the influx of adenosine into the cell and which leads to reduced activation of adenosine receptors. And reduced activation of adenosine A1 receptors lowers the threshold for epileptic seizures. On the other hand, the sink of adenosine drives transmethylation reactions. So if adenosine levels are kept low, we have an increased flux of methylation reactions through the methylation cycle, which can lead to an increase in DNA methylation. So that basically means if we increase adenosine therapeutically, we should have a very powerful way to block epileptic seizures. And we have shown this in a mouse model of pharmacoresistant epilepsy, where we inject a small dose of the excitotoxin canic acid into the hippocampus, which triggers focal status epilepticus. And after 3 to 4 weeks, the animals develop spontaneous epilepsy with electrographic seizures, which you can see here. A remarkable feature of this model is that those seizures are resistant to conventional anti-seizure drugs, such as carbamazepine. So if you give clinically used anti-seizure drugs, nothing happens. The animals continue to have seizures. However, as soon as you give an adenosine A1 receptor agonist, like CCPA, we get complete suppression of epileptic seizures. If we give an inhibitor of adenosine kinase to increase the levels of adenosine, we can also completely block those seizures. If we combine the adenosine kinase inhibitor with an adenosine A1 receptor antagonist, DPCPX, seizure activity is restored, providing proof that the adenosine kinase inhibitor acts via increasing adenosine acting on the adenosine A1 receptor. So this looks like a beautiful therapy, doesn't it? So unfortunately, like many things in science, this approach has a catch. We get very potent suppression of seizures, but at the same time, due to increasing A1 receptor activation, the therapy is highly sedative. So the animals basically start sleeping. So that's not practical from a clinical standpoint, because those systemically used drugs have too many cardiovascular and sedative side effects. So that means we need to find a different solution to make a smarter use of this powerful anti-epileptic potential of adenosine. In one of our earlier studies, we provided proof with encapsulated cell grafts, where we encapsulated adenosine-releasing cells into a semi-permeable polymer membrane, that the local release of adenosine close to a seizure focus is sufficient to robustly suppress epileptic seizure activity, as shown here in a rat kindling model of epilepsy. But importantly, seizure suppression in this model was not associated with sedation. So this bar graph shows spontaneous locomotor activity as a measure of sedation, and intraventricular adenosine did not affect spontaneous locomotor activity, whereas an anti-epileptic dose of the A1 agonist CCPA led to profound sedation. So this experiment provides proof of evidence that a local intervention to deliver or restore higher levels of adenosine in close proximity to a seizure focus is sufficient to block epileptic seizures. But now I want to really focus on epileptogenesis, which is the pathologic process that turns a healthy brain into an epileptic brain. So epileptogenesis is triggered by acute brain insults, which can be head trauma, stroke, infections, complex febrile seizures, or de novo status epilepticus. But remarkably, independent of the nature of the acute brain insult, a cascade of pathological alterations is initiated, which can constitute an epileptogenic injury, which finally leads to the onset of clinical epilepsy, which in humans can have a delay of months to years, and in rodents usually a few weeks. So during this time, brain circuitry is restructured on many different levels, and I don't want to talk about all of them, but I want to highlight in particular glial changes, which includes microglial activation and astrocyte activation and epigenetic reprogramming. So if you look at the pathology of temporal lobe epilepsy, we find a pathological increase in adenosine kinase, which implies a reduction in the ambient tone of adenosine. So on the left, you can see our mouse model of temporal lobe epilepsy, where we inject canic acid into the hippocampus. On the right, you see here in green massive astrogliosis, astrocytes stained with GFAP, which is associated with an increase in adenosine kinase, shown here in red, or in a DAB stain here in the epileptic hippocampus. So in the epileptic hippocampus, adenosine kinase levels are increased to 177% of normal, and this coincides with spontaneous electrographic seizure activity. So we find basically the same in human temporal lobe epilepsy. So this is material resected from patients with temporal lobe epilepsy that underwent the surgery to remove the epileptogenic focus, and likewise in temporal lobe epilepsy with hippocampal sclerosis, we see a massive increase in adenosine kinase in conjunction with astrogliosis. So this suggests that either astrogliosis or overexpression of adenosine kinase might be a direct cause for epileptic seizures. And to look into this hypothesis in more detail, we modified our epilepsy model and worked with a minimalistic model of epilepsy. So the model I've shown previously is a very complex model, which includes a lot of different histopathological alterations, including neuronal cell loss, granule cell dispersion, mossy fiber sprouting. So in this model here, we dissected out the role of astrogliosis and overexpression of adenosine kinase for seizure generation. So what we did is we injected a very small dose of canic acid into the amygdala, which triggers a focal onset status epilepticus, which we terminated after exactly 30 minutes with lorazepam. So 24 hours later, at the site of the original injury, we find local CA3 selective neuronal cell loss. So this is the acute injury, very small injury, just the CA3. If we then wait 3 weeks, we find massive astrogliosis with overexpression of adenosine kinase at the site of the original brain injury. So when we then implant electrodes into that area, we can record spontaneous electrographic seizures. And at that time, I had a very skilled postdoc surgeon who implanted electrodes simultaneously into different areas of the hippocampus, into CS3, CA1, and dentate gyrus. And as you can see here, the seizure activity is exclusively confined to CA3. However, as soon as he injected the A1 receptor antagonist DPCPX, seizure activity immediately spread throughout the entire hippocampus, leading to convulsive seizures, implicating that this area of focal astrogliosis is the origin of seizures, and that the adenosine system, the rest of the brain, is still intact and able to prevent seizure spread. To provide a direct cause that overexpression of adenosine kinase alone is sufficient for seizure generation, we generated a transgenic mouse, ADK-TG mice, to overexpress adenosine kinase just globally in the brain. And overexpression of adenosine kinase alone is able to trigger spontaneous electrographic seizures. And interestingly, overexpression of adenosine kinase also triggered a variety of comorbidities commonly associated with epilepsy, such as deficiency in working memory as studied here in a Morris-Watermaze paradigm. So then we looked in more detail at epileptogenesis in our transgenic mice. We engineered a second line of mice called FB-ADK-DEF, which are adenosine kinase deficient in the forebrain. So they have reduced expression of adenosine kinase in the forebrain. And then we repeated our intraamygdaloid canic acid injection. And here in the wild-type mice, you see the acute CA3 injury, CA3 selective astrogliosis, and overexpression of adenosine kinase and spontaneous seizures. When we did the same with mice that have reduced expression of adenosine kinase in the forebrain, nothing happened. So there's no astrogliosis, no overexpression of adenosine kinase, no seizures. But this is a very boring experiment because it's expected. So if adenosine kinase is reduced, we have more adenosine, which protects the brain. And you can see the protection actually in the CA3, which is not injured. So due to the acute protection, the animal did not receive the epilepsy triggering injury. So that's a boring experiment. But we then combined canic acid injection with an injection of the adenosine A1 receptor antagonist DPCPX. And this strategy allowed us to recreate the CA3 selective injury. And when we then waited 3 weeks, these animals were still protected. So this was our first hint that adenosine might have a novel anti-epileptogenic role, which at that time we couldn't understand. We couldn't understand why and how. So we did another experiment where we first injected canic acid into the amygdala to trigger the acute injury, which you can see here in those control images. And then after the acute injury, 24 hours later, we transplanted stem cells engineered to release adenosine into the hippocampal formation. And this is a GFP-labeled stem cell that has differentiated into a neuron. Here's another one. So those cells integrate into the hippocampal formation. And because those cells lack adenosine kinase, they are capable to release adenosine. So we then looked back at those brains 3 weeks later, and in our control animals, we find massive astrogliosis and overexpression of adenosine kinase. However, in those animals that received the adenosine-releasing brain implants, we find that astrogliosis was significantly reduced and adenosine kinase levels were almost normal. So what does this mean for seizures? What's your guess? Do those recipients of the adenosine-releasing cells develop spontaneous seizures? The answer is no. In over 100 hours of seizure recordings, we couldn't find any epileptic seizures in the recipients of the adenosine-releasing stem cells. So this is again an example that in this case, the local release of adenosine is sufficient to prevent epilepsy development. So this is again a very strong indication that adenosine is anti-epileptogenic. So, but still, the question was, how does it work? So we then started to look into epigenetics because at that time there was a growing body of literature coming up showing that increased DNA methylation or differential DNA methylation is associated with the development of epilepsy. So how can we link adenosine with this pathology of increased DNA methylation in epilepsy? So that's a study we published in the Journal of Clinical Investigation, where we basically nailed down the story that adenosine acts as a biochemical regulator of DNA methylation as a molecular basis for the anti-epileptogenic effects of adenosine. So what we need to consider is the following. So the adenosine-removing enzyme adenosine kinase exists in two isoforms, ADKS, which is expressed in the cytoplasm, But there's a second isoform, ADKL, which is expressed in the cell nucleus. So then we did a study where we generated baby hamster kidney fibroblasts to either lack adenosine kinase, so we used adenosine kinase knockout cells, and then we overexpressed either the cytoplasmic isoform or the nuclear isoform. And what you can see here is that overexpression of the nuclear isoform drives increased DNA methylation. And this is what happens if we increase adenosine kinase. We increase the flux of methyl groups through the transmethylation pathway, which then leads to increased DNA methylation. So if this is the case, then we should be able to block this process by the local delivery of high levels of adenosine. And what we did experimentally is we used adenosine-releasing silk. So this is true silk made from silkworms. So silk can be engineered as a drug delivery vehicle, and we engineered the silk in a way to release a defined dose of adenosine with a defined release kinetic for a limited time span of only 10 days. So if we increase adenosine, then we should be able to reverse the transmethylation pathway, which leads to an increase in S-adenosyl homocysteine, which is a known inhibitor of DNA methyltransferases. So with this strategy, we then should be able to block increased DNA methylation. And if increased DNA methylation plays a significant role in epileptogenesis, we should be able to prevent the development of epilepsy. So this is the design of our experiment. So we injected canic acid into rats systemically to trigger epilepsy. And we waited for 9 weeks until all animals had developed a first set of spontaneous convulsive seizures. So that's basically equivalent to the clinical diagnosis of epilepsy in a person with epilepsy. But in this model, epilepsy is progressive. So after the diagnosis of epilepsy, seizures get worse and worse and can be monitored for a long period of time. So we first looked at the brains at the time point when the animals had developed early epilepsy at 9 weeks after canic acid. And at this time point, we already find pathological overexpression of adenosine kinase and increased DNA methylation by increased levels of 5-methylcytosine. So then we implanted the adenosine-releasing polymer, which was engineered to release adenosine for only 10 days. And what we can see here is that the release of adenosine blocks the activity of DNA methyltransferase activity. So DNA methyltransferase activity is increased in the epileptic brain during active adenosine release from the polymer. DNA methyltransferase activity is almost silenced. So then we looked at global DNA methylation levels. In the early epileptic brain, we already find increased hippocampal DNA methylation. However, during active adenosine release, DNA methylation levels went back to normal. And excitingly, this effect was maintained even after cessation of adenosine release from the polymers. So we basically have a long-lasting epigenetic effect after transient adenosine therapy. So then we really looked into epilepsy development in our animals, and that's the design of the experiment. We started our treatment at the development of early epilepsy, 10 days of adenosine release, and then we continued to monitor the animals for a total of 21 weeks. So what we find is the following. So all animals at 9 weeks already had early stage epilepsy with up to 4 seizures per week. And so then we did our implantation, either adenosine releasing silk or silk that doesn't release anything, or sham treatments. And what you can see here is that there is a progression in epilepsy development in all of our control animals. However, the animals treated with adenosine showed almost complete suppression of seizures during active adenosine release. And after cessation of the adenosine release, the seizure profile stabilized. So the seizure phenotype was basically frozen at the level we had prior to the polymer implantation. So that means that a transient dose of adenosine prevented epilepsy progression long-term for at least 3 months. What we also found as an independent experimental measure, that the transient release of adenosine prevented the sprouting of mossy fibers, which is a pathological hallmark of the epileptic brain. So we then looked at gene regions regulated by methylation during the course of epilepsy development, and those that can be reversed by adenosine, and did bisulfite sequencing to identify distinct changes in methylation sites. And eventually we came up with a long list of epigenetically regulated targets. And interestingly, among the top targets that we found targets that are all proteins in the cell nucleus, like polymerases, RNA polymerases, DNA polymerases, and zinc finger proteins. We found a lot of proteins that directly interact with DNA. So therefore, we think that this is a regulatory mechanism that acts very globally on the general homeostasis of the DNA methylome and transcriptome. So this brings us to the adenosine kinase hypothesis of epileptogenesis. And what this boils down to is basically that after a trigger for epilepsy, we initially have a surge in adenosine as an acute protective mechanism. But then after a latent period, we get a lot of maladaptive changes, which include astrogliosis, overexpression of adenosine kinase, and adenosine deficiency, which then triggers the hypermethylation of DNA, which is needed for the maintenance and progression of epilepsy. And if we block hypermethylation of DNA with adenosine therapy, we are able to interrupt this cycle and are able to prevent the progression of epilepsy. So that basically means that adenosine kinase is a very attractive target for drug development, and specifically for the prevention of epilepsy. So adenosine kinase is a relatively small enzyme, which is encoded by a huge gene. So the gene in humans is over 500,000 base pairs, and it only has 11 exons with a coding sequence of 1.1 kilobases. So this gene is so big that it takes 8 hours to transcribe. But adenosine kinase is very highly conserved in evolution. It's an ancient protein derived from ribokinases. And what happens if the enzyme is lacking? So this is some early work from my lab where we knocked out adenosine kinase in the mouse. And in this study, we made the discovery that adenosine actually plays a very important role in the transmethylation cycle. What we find in these adenosine kinase deficient mice is developmental retardation and stunted growth, and most importantly, hepatic steatosis. So those livers are yellowish and they are filled with fat. And the reason is because the liver is the #1 transmethylation organ in the body. So the liver produces all the methylated lipids like lecithin, for example, and liver is also the organ with the highest expression levels of adenosine kinase. So interestingly, in the very same year, another study was published where they knocked out the adenosine kinase gene in Arabidopsis. And remarkably, those Arabidopsis plants that lack adenosine kinase have exactly the same phenotype as our adenosine kinase knockout mice. They're characterized by stunted growth, and they identified the same defects in the transmethylation pathway. So this is a really nice example of how science works. So you do one work in mice and another work in plants, and you get exactly the same results. Subsequently, adenosine kinase deficiency has been identified in humans, and those humans are also affected by stunted growth and developmental retardation, a different shape of the head, and the authors of those studies also identified the same kind of transmethylation defects. So if we want to exploit adenosine kinase therapeutically, we need to consider the existence of those two isoforms. The isoform ADKS, expressed in the cytoplasm, is basically responsible for non-cell autonomous effects of adenosine, which basically means regulation of the tissue tone of adenosine and control of adenosine receptor-dependent effects, which are important for seizure control, but also for side effects. On the other hand, ADKL in the cell nucleus mediates cell autonomous effects, which have a role for cell proliferation, cell fate, all those epigenetic effects, which I've outlined previously, which are important for anti-epileptogenesis. So therefore, the therapeutic goal for drug development is to develop adenosine kinase inhibitors with a known activity on ADKL, preferably compounds that are more potent for ADKL than ADKS. With those compounds, we should be able to capitalize on the epigenetic effects of adenosine while minimizing systemic side effects. So adenosine kinase inhibitors have already been used in clinical drug development in the early 2000s, but they largely failed because they were designed for the constant chronic use for the indication seizure suppression, chronic pain, and chronic inflammatory diseases. The long-term use of those agents was just not possible because of sedation, cardiovascular side effects, and liver toxicity. However, since we have shown that the short-term use of adenosine is able to have long-lasting epigenetic effects, there might be a new future for adenosine kinase inhibitors if they are just used transiently to capitalize on epigenetic effects. And this is a proof of principle study in our intrahippocampal epilepsy model, where we inject canic acid into the hippocampus to trigger epilepsy. And then we gave just a 5-day treatment with the adenosine kinase inhibitor 5-ITU during the latent phase. And then we looked at seizure development in the animals at 6 and 9 weeks. And what you can see here is that in over 50% of all animals, we were able to completely block epilepsy development. And this is a very significant result from for a clinical utility perspective. So that means that a transient dose of an adenosine kinase inhibitor is actually capable of preventing epilepsy. So now I want to switch gears a little bit because studying adenosine kinase in the brain led us into the area of cancer. So adenosine has been a hot target for cancer therapy, in particular the adenosine-producing enzymes, because it has been known that cancer cells are capable to release adenosine into their microenvironment, and adenosine-releasing cancer cells have tremendous advantages because adenosine is an immune suppressant and it promotes angiogenesis. So if a cancer releases adenosine, it blocks detection by the immune system and promotes its own nutrient supply through angiogenesis. So, but what has not been realized previously is that the adenosine system is highly compartmentalized. So on all clinical initiatives to make use of the adenosine system therapeutically, people have looked almost exclusively at the adenosine producing cascade involving the enzymes CD39 and CD73. And they have clinical trials using antibodies directed against CD73 to reduce the formation of adenosine. But the system is a lot more complex because there are a lot more extracellular enzymes involved, and extracellular adenosine also depends on intracellular adenosine metabolism. And so this led us to the submission of a cancer grant to the NIH, where we kind of dissect out the role of the different components of the adenosine system and divide the system into an extracellular component, an intracellular component, and a component within the cell nucleus, where we are dealing with the epigenetic effects of adenosine. So that means within the adenosine system, we have a lot of promising and potential targets for anti-cancer therapy. The challenge is to put all those pieces together and to look at the entire plurinome in a comprehensive manner to design the best possible treatments for cancer. So what we found is, and this is important for cancer, is that adenosine kinase plays a big role in development and plasticity of cells. So we found that there's a dramatic shift in the expression of the nuclear isoform of adenosine kinase, ADKL, which is the long form in those Western blots. So what you can see on those Western blots is that in the brain, proliferative plastic cells have high levels of ADKL, and in the adult brain, the ratio shifts to a dominance of ADKS. So that means in brain development, ADKL is associated with plasticity and proliferation. And the hypothesis is that in cancer, we have basically a de-differentiation of the adenosine system, where high levels of ADKL actually may promote a more plastic phenotype of cancer cells and may promote cell proliferation. So in the brain, we found using knockout mice, so this is a knockout mice where we deleted adenosine kinase from dentate granular neurons in the hippocampus. And those are mice where we overexpress adenosine kinase, the L form, throughout the entire hippocampus. So that's a normal hippocampus, but those mice were engineered to overexpress the nuclear isoform of ADKL. And then we looked at baseline neurogenesis in those mice. And in line with the role of ADKL in promoting cell proliferation, we find increased cell proliferation in the hippocampus of those ADKL transgenic mice, suggesting that ADKL may drive cell proliferation under baseline conditions. And to directly look at the relationship of ADK expression with cell proliferation, we used our engineered BHK cells that either lack adenosine kinase or overexpress either ADKL or ADKS. And then we looked at cell proliferation, and what you can see here, that overexpression of ADKL leads to a dramatic increase in cell proliferation, whereas lack of ADKL and overexpression of ADKS block cell proliferation. So therefore, we need to distinguish between the forms of adenosine kinase, and that means that adenosine kinase, and in particular ADKL, is also a potential target for cancer therapy. And looking at human breast cancer, we actually find that in tumor samples labeled with the T here, we find high levels of ADKL expression, which associates with cancer in a total of 46 breast cancer patients. So therefore, I teamed up with our colleague Ken Jacobson at the National Institutes of Health to develop a new series of adenosine kinase inhibitors with the goal to develop novel inhibitors that are selective for ADKL. And our first set of compounds shows that we already found several new inhibitors that are more potent than the 5-ITU we used previously in our mouse model. And I don't want to go into the details here. So we also developed cell-based drug discovery platform to directly screen for the epigenetic activity of those new compounds by their capability to reduce global DNA methylation levels in cells that only express ADKL. And this is one of our lead compounds, 4203, which leads to a profound reduction in DNA methylation in ADKL expressing cells. So that basically means with adenosine, we truly have a very translational molecule in hand that we can use therapeutically to increase the activation of adenosine receptors to affect methylation reactions, which is of potential use for a wide range of pathological conditions. And we can affect adenosine by the use of gene therapy, stem cell therapy, ketogenic diets, yes, those silk-based brain implants and adenosine kinase inhibitors. And with this, I would like to thank you all for your attention, and I wish to thank my research team at Rutgers and my collaborators and funding sources. So thank you very much, everyone, and I'm happy to answer any questions.
Excellent. Thank you, Detlev. That was a remarkable review of your work on adenosine and beautiful slides as well. I'd like to, someone is still coming into the waiting room. People are still joining the meeting. I think this person missed the talk, but so I received one question in the chat from our new professor, Sally Seraphin. So I'll ask that question first and then open it up to the floor. But she was wondering, in some of the intro neuroscience classes, there's discussion about using colostomy, split brain procedure for treating epilepsy. And she wanted to know if maybe adenosine-based therapies could eliminate the need for that kind of a brain surgery for epilepsy.
Yes, no, this is, no, that's a very good question. So there are a couple of surgical procedures in place to, yeah, basically treat epilepsy through a very drastic measure. So far, we have seen that adenosine augmentation works in every epilepsy model we have looked at. So the challenge is to really make therapeutic use of adenosine and at the same time alleviate side effects associated with systemic administration. So in order to suppress seizures, we need to increase the activation of adenosine A1 receptors, which is challenging if we do it systemically because of cardiovascular and sedative side effects. So in those split brain procedures, yeah, we are dealing with hemispheric epilepsies or generalized epilepsies that usually don't have a clear focal origin. So those epilepsy would need to be treated possibly brain-wide. And to do this chronically with a brain-wide adenosine augmentation approach might still be challenging because of sedative side effects. So I know that allosteric adenosine A1 receptor agents are in development. This might be an option to get more selective drugs, but currently there are no readily translatable options, I'm afraid.
Interesting. Let me open the floor to our remote people who are there. If anyone wants to make a comment, that'll make it easier for me to kind of keep track here. Is there anyone who's piping in remotely that wanted to ask a question? All right, well, keep it in mind. I'll offer it again in case you couldn't find your unmute button. How about people in the class? Did you have some questions? I know we talked about some of his work last week. Yes.
So adenosine clearly has some very broad effects. Could disruptions in that translate to symptomological patterns that don't really fit a diagnosis, like neurodegenerative diseases that don't seem to clearly fit the models of Alzheimer's or Parkinson's neatly?
Could you hear that, Detlev?
Yeah, no, we have actually looked at both Alzheimer's disease and Parkinson's disease. So what we find in those neurodegenerative conditions is they also share astrogliosis with epilepsy, and we also find overexpression of adenosine kinase and adenosine deficiency. We actually looked at adenosine levels in the cerebrospinal fluid of Alzheimer's and Parkinson's patients, and we found a massive reduction in adenosine in those patients, implicating that adenosine deficiency plays a major role in the pathology of those diseases. And it makes sense if you have a reduction in adenosine, which acts as an endogenous neuroprotectant, neurons are more likely to die.
Yeah, I think her question was, could there be kind of an unusual, difficult to diagnose thing that might be related to an adenosine dysregulation? Was that your question? And I think the answer is yes, but probably we've developed names for all of those things, and we never knew that there might be an underlying adenosine dysregulation, but I'll let you answer that question.
Yeah, no, that's a very good point, because diseases have been defined historically over 100 years ago and got a name based on the primary symptom. But it could be, and that's a kind of a provocative thought, that we should redefine diseases based on the underlying biochemical or genetic disturbance. A classic example is, for example, cancer. When we talk about breast cancer, prostate cancer, liver cancer, and so on. But this is a relatively superficial approach. It's just based on the location where the cancer comes from. Another approach would be to categorize cancers across their molecular signature. For example, to distinguish between IDH1 mutant cancers and non-IDH1 mutant cancers, and based on the molecular signatures to group cancers in a different way to understand them better.
Interesting. Other questions from the students that are here? Donovan, yes.
I was really interested about
You're gonna have to yell a lot louder.
sorry. I was really interested about the adenosine releasing silk. Can you please speak a little more about that?
The question was talking a little bit more about the adenosine releasing silk.
Yeah, so now if you think, you can use silk as a biomaterial to turn almost into everything. So this was in collaboration with David Kaplan, who is the director of the biomedical engineering department in Medford. And he's kind of a silk guru, and he can turn silk into translucent artificial cornea. So he once had a grant from the CIA to produce a paper made out of silk. So you can write on the silk paper, and when you no longer need it, you can eat it and it leaves no trace. So you can turn silk into almost everything, and most importantly, into a drug delivery vehicle. So the way the drug release works, if you just encapsulate a compound like adenosine in a silk membrane, you always get a logarithmic release profile. Now you get first a burst of release, and then the release slows down. So that's the normal release kinetics. But if you combine several logarithmic release profiles that each have a different kinetic in one construct, you can kind of even out those logarithmic release curves and get a more linear release. So the way those implants were constructed, we first encapsulated adenosine as such into silk-based microvesicles, which we then embedded in a 3D silk scaffold. And then this 3D silk scaffold was coated with alternating layers of adenosine and silk, and so on. And then we added additional capping layers of silk. So, so those constructs are quite complex, and in the end, if you do it right, you can get a nice linear release profile.
Thank you. Other questions?
I'll ask one. This is David Ruskin online. Detlev, I know you've spoken today about the two main kinds of adenosine kinase, but in our discussions, I know you've brought up that there are actually maybe many more. Could you say a little bit about that?
That's a very good question. That's really exciting. So there are two main forms. That's ADKL and ADKS. But since everything is sequenced these days, and with all these massive scale RNA-seq approaches in human, a total of 14 different splice variants of adenosine kinase have been found. So presently, it's completely unknown what those different splice variants do, what their role is. If they, so what we've seen is we have never found any of those prominently expressed in healthy tissue, but there's possibly a likelihood that some of those forms are expressed in pathological tissue. So the most interesting findings from our lab is, so we did a panel of human hepatocellular carcinoma samples. And in those liver cancers, we did Western blot analysis. And in most of those human liver cancers, we found more than 2 adenosine kinase bands. But not in healthy tissue. So it could be that some of those forms have more a pathological role than a role in the healthy tissue or specific developmental functions. But it's probably something to keep me busy for the next 10 years or so.
Thanks.
Yeah, I was like, what do you mean that's good news? Now we have a whole nother zoo of things to worry about, so.
Yeah.
Other questions from the floor or from the remote people?
Yeah, I have a question.
Yeah.
You mentioned briefly depression. Can you expound on how depression has something to do with seizures and epilepsy, and whether or not epilepsy exacerbates depression and vice versa? And also, what is the role of adenosine in this whole depression phenomenon?
Yeah, that's a very good question. So it is known that depression is a major comorbidity of epilepsy, and so adenosine deficiency might actually be the cause for depression. And so Phil Hayden at Tufts has done very beautiful work on looking at the role of adenosine in depression. And so it is known clinically that 1 night of sleep deprivation is one of the most potent and most rapidly acting measures to treat acute clinical depression. This sounds almost strange, but one night of sleep deprivation helps for acute depression. So one night of sleep deprivation triggers a massive increase of adenosine in the brain, which might actually be the reason why this treatment works. And using an elegant set of molecular tools, Phil Hayden has nailed down the story that adenosine actually has an antidepressant role. Now there's also crosstalk between adenosine and dopamine. So the adenosine receptors from heterodimers with dopamine receptors, and also metabolically, adenosine might regulate the biochemistry of dopamine, because the major dopamine removing enzymes is catecholamine methyltransferase. So that's a methyltransferase which inactivates dopamine. And because transmethylation is regulated by adenosine, it could be that adenosine directly can regulate the availability of dopamine through this pathway. So there are very complex interactions between adenosine and the dopamine system, which can possibly explain a lot of those comorbid conditions.
So Detlev, related to that question, Professor Seraphin just typed in the chat. So conversely, can caffeine cause depression? And I think I know, I have my answer to that, but I'll see what you think related to that.
I don't really know.
So I would say that I think caffeine actually has been shown to help depression, but I think it's because it acutely blocks adenosine receptors, which causes a general upregulation of adenosine tone when the caffeine isn't there. So I don't think there's any evidence that caffeine causes depression, just like there's no evidence that caffeine is making epilepsy worse, because it's present for that acute amount of time.
Yeah, I completely agree. Yeah. Mm-hmm.
I'll just jump in here as well, if I may, in terms of caffeine. So the thing with caffeine is it depends on the dosage, and it also depends on the subject and whether or not the subject is a person who drinks a lot of caffeine and has been drinking a lot of caffeine for many years, because people do develop some type of resistance. So it depends on the subject, it depends on the dosage, but caffeine can also cause side effect of sleep deprivation as well. And there might be some indirect interaction between caffeine and depression, probably most likely through the sleep deprivation aspect of this.
Yeah, that would make sense. Yeah, yeah. Mm-hmm.
Good, thank you. Thank you, Professor Blaze. Other questions from the floor, from our remote people? I have two things that I wanted to point out, because every time I see you give a talk, I think of something else. And I know you recently moved to a neurosurgery department, and I never knew that, I knew that adenosine was given for tachycardia. I didn't know it was given for neurosurgery to detect the bleeding artery in the brain. So did you just find that out more recently, working more closely with these neurosurgery colleagues? Because I didn't know.
Yeah, I didn't know either. So when I was talking with them about adenosine, they told me, yeah, we use it all the time.
That's so funny.
Yeah.
Okay, because I thought it was used very rarely in case someone had a severe heart event on the operating room table. This seems like a fairly common use of adenosine in the operating room.
Yeah, common in the sense as common as severe bleeding in the brain is. So if there is really severe bleeding that they don't know where it comes from and they have to stop, then they have to give caffeine. It's still kind of an emergency treatment.
Mhm. Okay. And I also wanted to really, emphasize, I mean, it's just beautiful how your work has unfolded this kind of strategic story of trying to understand adenosine, and that your work has kind of come full circle back to some of the very initial topics that you were interested in the liver with adenosine kinase and the methylation pathways, and that biochemistry is now something that you're bringing to the fore with these neurological conditions and cancer. So very exciting, kind of, and the relationship to plants, which we need to
Yeah.
kind of related to everything else on the planet.
Yeah.
So just give one final call for additional questions. Any final thoughts for anybody? Any final thoughts that you wanted to share, Detlev?
No, I think the take-home message is that adenosine is the molecule of life.
Yes, right. So thank you so much. So I hope that my students have been able to see why, I've been working for so many years on adenosine and think it's so important. And I'll ask them to please help me in thanking you again and thanking you for allowing us to record it, because there's a lot of content that I'm sure people will want to look at again. So thank you so much.
Yeah, thank you very much. Yeah.
Talk to you soon, and thank you everyone for joining.
Okay, thank you.